ever wondered how insulin is made? Insulin is made by a technique called DNA recombinant technology. And this is the topic of today. Recombinant DNA is a form of artificial DNA that is made through the combination or insertion of one or more DNA strands. In genetic, recombinant DNA technology has many uses. In agriculture, growing crops of your choice. In pharmacology, artificial insulin production. In medicine, as in vaccination. Other uses as in growing plants. About advantage of recombinant technology: it's unlimited utilization, cheap, resistant to natural inhibitors. But about disadvantage of recombinant technology: it affects the natural immune system of the body, also can destroy natural ecosystems that relies on organic cycles. Cloning: the term clone means exact copy of the parent, a duplicate or a look-alike carrying the same genetic signature or genetic map. Molecular cloning is carried out in vitro, where a specific fragment of DNA is isolated from an organism, donor, and introduced into a plasmid that replicates in a host cell, making multiple copies of that DNA fragment. Plasmids that are double-stranded DNA, usually circular, mostly found inside certain bacterial species. Plasmids, also known vectors. Vectors also include the viruses known as bacteriophage that use the bacteria as their host to replicate. Types of vectors used: plasmids, bacteriophage وcosmids والBACS والYACS. Steps involved in molecular cloning using recombinant technology. First, DNA from donor is isolated and purified. Second, restriction enzyme endonucleases generate fragments of purified DNA by cutting the DNA at reconjugation site. مهم جدا نعرف ان لكل restriction enzyme في reconjugation site specifically. Third, the fragments are inserted or pasted or spliced into plasmid. Plasmid transferred to host cell. As the host cell replicates, recombinant molecules are based on the progeny known as clones. Finally, cloned DNA can be recovered, analyzed from the host cell after processing. This show figure restriction enzyme action of E. coli. This also show plasmid and donor DNA. Plasmid as a vector with a cleavage site, donor with a cleavage site. Cleavage site by E. coli endonucleases in both donor DNA and the plasmid. اتكون لي حاجه اسمها الهايبرزينيشن. الهايبرزينيشن دوت بالدي ان اي لايجيز هيكون لي الريكامبينانت بلازميد. At specifying the cloning insulin AB chains using recombinant DNA technology. DNA strand with a specific nucleotide sequence for insulin chain A and chain B. Unrevealing the strand of the DNA of chromosome expose the nucleotides coding for the AB chain. يعني بيحصل separation للتو strands بتوع ال DNA. Third, restriction endonucleases cut the insulin gene fragment by recognizing the particular nucleotides. Finally, the synthetic A and B chain genes are then separately inserted into the gene for bacterial enzyme beta-galactoside, which is spliced into and carried by the plasmid. The recombinant plasmids are then introduced into E. coli cells. The insulin gene is expressed as it replicates with the beta-galactoside in the cell undergoing mitosis. كل ما يحصل انقسام ميتوزي للبلازميد هيتكون لي انسولين من النوع A and B chain insulin protein. The protein which is formed consists partly of beta-galactoside joined to either A or B chain of insulin. The A and B chain are then extracted from the beta-galactosidase fragment and purified. Clone selection. Clones carrying the recombinant clones are differentiated from normal clones using clone selection. As in case of recombinant insulin, 3 types of E. coli clones grow on agar. normal clones وclones with A chain insulin protein وclones with B chain insulin protein. There is a certain antibodies could be added to the agar that would inhibit the growth of normal clones with normal plasmid inserts. ده هتعمل لي ايه؟ هيحصل growth only لل A chain insulin protein وال B chain insulin protein. Finally الستام cells عن 3 أنواع من الستام cells: embryonic stem cells و adult stem cells و mesenchymal stem cells. Embryonic stem cells have the ability to differentiate into all cells, early showing potential advantage, but ethics and its ability to cause cancer limit its usage. Second, adult stem cells, hematopoietic stem cells have been in use for some time now. They can be used to synthesize blood cells, but recipient has to be immunocompromised. Third, mesenchymal stem cells can be obtained from cord blood or umbilical cords. They have the ability to be programmed using certain growth factors to give a specific cell. Has a great potential for various utilization in future. Thank you.